Hello everyone, my name is Erin Lat.
Hello, my name is Fu Ling.
Hey, my name is Narma.
Hello, my name is Natasha Anak Jayong.
Hi, my name is Vivana Jessie. First of all, we label all the test tubes so we don't get confused. Next, 4 ml of distilled water was placed in 7 test tubes using micropipette, while the 7th one is for a blank solution. Next, 1 ml of Bradford reagent was added to each test tube, including the blank solution. Then 50 microliter, 100 microliter, and 200 microliter of BSA protein standard were added to each test tube in duplicate except for the blank solution. After that, all the solution is mixed well by using a bench mixer. The solution in each tube then was placed in a cuvette for a spectrophotometry analysis. Next, each cuvette will undergo spectrophotometry to measure the absorbance values, starting from the least concentrated sample. The blank, which contains no protein, is used to zero the spectrophotometer before taking any readings. Moving to part 2. The process is similar to part 1, except this time we'll be using the unknown protein standard solution A, B, and C. Each sample will be measured for its absorbance at 595 nanometer using the spectrophotometer. As before, the spectrophotometer is blank before starting, and the absorbance readings for each unknown sample are then recorded. This table shows the absorbance value obtained when different volume of BSA protein standard were added to the Bradford reagent and measured using a spectrophotometer at 595 nanometer. Test tube 1 with 0 microliter BSA acted as the blank, giving absorbance of 0. This is used to zero the spectrophotometer. For the 50 microliter BSA, the average absorbance is 0.180 with a standard deviation of 0.0799, indicating a slight variation between the duplicate readings. For 100 microliter BSA, absorbance readings are more consistent, with average of 0.266 with a lower standard deviation 0.005, showing the readings are very close to each other. Next, for the 200 microliter BSA, the average is 0.289 and standard deviation is 0.00141, indicating excellent reproducibility. From this graph, we can see when the volume of BSA increases from 0 to 200 microliter, the absorbance also increases. This shows a positive correlation, meaning that higher protein concentration leads to stronger absorbance readings. This is because the Comasi brilliant blue dye binds to protein and change color, which we can measure at 595 nanometer. Each point on the graph represents the average of two absorbance readings for each BSA volume. There's also a red arrow bar at 50 microliter showing the standard deviation of the two readings. The large arrow bar suggests that there were more variation between the two results at this point. In contrast, at 100 microliter and 200 microliter, the measurements were very consistent, with almost no variation. Overall, the graph forms a standard curve that we can use to estimate the protein concentration of unknown sample based on their absorbance. Moving to part 2. To determine the concentration of unknown protein solution A, B, and C, we use the formula derived from the BSA standard curve, which is y equals to 0.02x. Using this formula, we obtain the following result. Solution A has a concentration of 14.80 microgram per milliliter. Solution B is 15.95 microgram per milliliter, and solution C is 18.25 microgram per milliliter. Next, this is the graph of protein concentration versus absorbance. As we can see, there's a linear relationship shown by the equation y equals to 0.02x with r squared equal to 1, which means the data is highly accurate. The linear trend shows an increasing trend as the absorbance at 595 nanometer reading increases and the concentration of the protein also increases.
Here are the new learnings that I gained after doing the protein analysis using a spectrophotometer. The first and foremost is that I learned how spectrophotometry works based on the principle that proteins absorb light at specific wavelengths, which is around 280 nanometers due to aromatic amino acids. This gave me a clearer picture of how light absorption correlates with the concentration. And not only that, I realized that even the slightest mistakes in pipetting or mixing can affect the accuracy of the readings. This says that proper sample handling and consistent techniques are crucial for valid results.
The challenges faced during conducting this experiment are: first, difficulty in accurate pipetting and sample preparation. Second, minor error lead to inconsistent adsorbent readings. And last, initial confusion interpreting the standard curve and calculation.
Lastly, suggestions for improvements are: provide more practice time with micropipettes before the experiment. Besides, include a pre-lab video or demo to explain spectrophotometer usage. Moreover, use more replicas per sample to improve data accuracy.